Krebs cycle. So in this video, we're going to be looking at both the Krebs cycle, which is going to be the second phase of aerobic respiration, and we're also going to be looking at the third phase as well. We're just going to squeeze them both in one video. So the third phase, oxidative phosphorylation, comprises of two others, the electron transport chain and ATP synthase. So if I were to put it here just as a reminder, so aerobic respiration, we mentioned in the previous videos that there were three phases, right? So the first one was glycolysis. So we finished that one. Now we're on our second phase, our Krebs cycle. And our third phase is oxidative phosphorylation. And that's split up into two subcategories, our ETC, the electron transport chain, and as well as ATP synthase. So let's tackle this one right now. So you remembered from the previous video, we ended off with two pyruvates, okay, at the end of glycolysis. And provided that we have enough oxygen, we assume that that's in plentiful amount in order to create 2-acetyl-CoA, or coenzyme A. So let's say this pyruvate is illustrated by these three carbon molecules for simplicity. We're going to make that so that one pyruvate is going to make 1-acetyl-CoA, and then I'll just say at the very end that just happens twice. So then we'll just do times 2, because obviously you can see that we had two pyruvates creating those 2-acetyl-CoAs. Okay, so we'll just tackle one for now and then just duplicate it. So our first pyruvate over here, to make that 1-acetyl-CoA, it's going to give off a CO2, because Krebs cycle is simply an oxidation reduction reaction. So the carbons are going to be oxidized, okay? This carbon is going to be oxidized to harvest more electrons. When we get to the electron transport chain, we're going to be looking at a few chemical equations where I can kind of go more into depth about oxidation reduction, okay? So when that carbon dioxide gets released, we're left with 2, but then this 2-carbon molecule is going to then bind with a 4-carbon molecule that's floating around, okay? So let's have this one floating around here. And the name of this one is oxaloacetate. And it's pre-existing, okay? So this pre-existing oxaloacetate then meets and combines with that acetyl-CoA. Then they're going to form a, obviously 4 plus 2 gives you a 6-carbon molecule. So let's have that over here, and the name of this one is citrate. Citrate. What's going to happen next? Well, surprisingly enough, carbon dioxide once again gets removed, okay? There is oxidization happening, and there's 2 CO2s that get released. This helps to ensure that any ADPs that are floating around are able to bind with the inorganic phosphates in order to create that ATP. Only one ATP. So this is very inefficient, and it's not a lot of ATP, but at least it's something, okay? What else happens when that carbon gets fixed? Well, FADs turn into FADH2s. FADH2 is a high electron carrier. High electron carrier. And this is one molecule that is going to be absolutely crucial for the next stage, okay, which is the oxidative phosphorylation. Then that carbon fixation also ensures that 3 NAD pluses can turn into 3 NADHs. Again, this is also a high energy carrier. electron carrier. Okay, and again, same thing with the FADH2, right? The NADHs are going to donate those electrons that it's holding on in order to generate ATP for the next phase. Now you're probably wondering, how did it get these electrons, right? Same thing with FADH2. How did it get these electrons? Again, we're not going to get into depth. There's a bunch of different enzymes that, like isocitrate, malate, some other enzyme like glutarate, okay? They help to ensure that they give those two electrons to the FADH2 and the NADH during their construction, and varied like protons as well to help make that NADH and FADH2. But again, we don't need to get into that deep of detail. Now, while all of that is happening, there is going to be some regeneration. Okay? Because remember, this had 6 carbon molecules. We lost 2 from the carbon fixation, so we only have 4 left. So we are back to oxaloacetate again. And then that's going to meet another acetyl-CoA, and then the cycle just repeats. And like I said, this happens twice, right? So technically, instead of just making 1 ATP, you've actually made 2 ATPs. Okay, so 2 ATPs were generated. Same thing with the FADH2s. Instead of 1 FADH2, we've made 2 FADH2s. As well as the NADHs, instead of 3 NADHs, we now have 6 NADHs. Now let's get to oxidative phosphorylation. So regarding our electron transport chain, which is what needs to happen first, why do we need to go through this process? Like I said, it's to generate ATP. The electron transport chain is going to help us to produce a proton gradient using energy from the electrons, from those 2 things that we just talked about, right? The NADH that we made in the steps before, and the FADH2s, right? All right, so I'll just write that over here. To generate ATP, way more ATP. Like I said, the electron transport chain, it's to produce a proton gradient, right? This is for proton, a proton gradient using energy from the electrons, from the high energy carriers, NADH and FADH2. And we're going to see that the protons are going to be in the form of H pluses, because technically it is a proton, if you remember from chemistry. And what's that going to do, though, having that big proton gradient built up? It's for our ATP synthase that I mentioned earlier. That ATP synthase ensures that the proton gradient serves as a proton motor force. To synthesize ATP. I'll just put synth, okay, to synthesize ATP. And it's a lot, about 30 to 32 ATP. And please remember, the important part about oxidative phosphorylation is the first part in its name, right? Oxidative, which means it needs oxygen to produce all this ATP that we're going to be doing. So those 2 electrons that are going to be floating around are going to reduce oxygen in order to make H2O as our product at the very, very end. All right, so let's get into nitty-gritty about reduction and oxidation, okay? Please see the video links in the description for balancing half reactions, as well as understanding oxidation numbers, as well as identifying redox reactions. So once you've watched those, you can come back here, okay? So pause the video right now, watch those videos, and then come back. So now that you know that whenever there is a transfer of electrons, it doesn't just disappear. Whenever it gets oxidized, it's going to be reduced as well. So it's going to be given somewhere else. So let's have a breakdown below. First, I want to emphasize NADH. let's use this as an example. So that NADH turning into NAD plus. I'm going to be showing you this reaction, as well as I'm going to do a second one as well, talking about oxygen turning into H2O. Okay, both of those are going to be shown in this next diagram. But before I show it, I want to clarify the oxidation reduction though. And with the help of those videos, hopefully it should be a little bit more clear right now regarding oxidation numbers. So you have an oxidation number here of +1, whereas it started as a reactant with 0. So if your oxidation number here is 0 and it's a +1 over here, is this oxidation or is this reduction? This is obviously oxidation because there is an increase of the oxidation number. So I'm going to put oxidation here. And if you saw the video for the balancing of the half reactions, especially in the acidic solution specifically, H2Os, if they appear on either side, how do we balance them? You ensure that H pluses are added on the side, on the opposite side, to make sure that they are balanced. But to ensure that the charges are balanced, you have to add the electrons in. Now, I'm not going to give you the full breakdown of the waters and then the H pluses on either side and the electrons on either side. Okay, I'm not going to go through that process. You should already be adept by now based on watching that video. I'm just want to show you the end product. Okay, the end product ended up with an H plus on this side, as well as these 2 electrons. And if you look at the oxidation numbers, this one, like we said, was 0. This one was a +1, but then there's another +1 with a proton, the hydrogen ion, and then there was 2 electrons in order to cancel them out. So then the oxidation number here is 0 as well. So we're good. Another way to identify that this was oxidation, rather than just looking at the oxidation number, was which side are the electrons on. The electrons were on the product side. That means that they were given away. They were produced, okay? It wasn't taken. Taken would be on the reactant side. If it's produced or given away, it's going to be on the right side. So the electrons were given away, so they were lost, which is oxidation. Very similarly, electrons run into the oxygen and break it apart. Well, why is it reduction? Let's have a look here. Well, you know oxygen is O2. It's a diatomic molecule, right? It cannot exist by itself as just O, okay? It needs to be bonded with another O. Well, in order to break it up, you would need 2 electrons. So if I were to create my chemical equation here, what are reacting? Like I said, electrons are going to be reacting with the oxygen. But this O2 got split in half, so I'll just put half of oxygen. So those 2 electrons were used, but what else is floating around and gets added into the equation? Hydrogens as well, hydrogen ions. Now, if you don't know why oxygen and hydrogens are very, very good at combining and reacting, see the video link in the description for electronegativity. Oxygen typically has a very, very high electronegativity, and hydrogens is very, very low. So they hydrogen bond, but again, we don't have to get into the specifics, just know that they like to interact. So obviously, oxygen, hydrogen, what do you think it creates? Obviously, H2O, which is what you all know, the hydrogen monoxide, also known as water. Now this doesn't look balanced though, so how do we balance it? Well, we have 2 hydrogens here, we only have 1 there, so let's add a 2. Are we balanced? Yeah, it looks pretty balanced, right? We have half of 2 oxygens, so this is just 1, and we only have 1 oxygen over there, right? So we're good. And let's look at our charges. 2 electrons, which means this is a -2, -2. 2 H pluses, so that would be a 2 plus, so then they cancel out. So I think we're good. Just remember. But if you want to make this a little bit more clean, where you don't want to see this half in front of it, you can just make this an O2 if you want. Because remember, it got split, right? The O2 got split into its oxygen ions, but then obviously the 2 electrons and the hydrogens get involved, so it's going to turn back into that O2. So if you want to do it that way, you could have an O2 over here, okay? But obviously we have 1 oxygen there. How do you make 2 oxygens? You got to times 2, right? So if you times 2, everybody here gets times 2 as well. Okay, so this is times 2, okay? If you're going to times 2 on that one, that will give you 4 electrons. If you're going to times 2 on that one, that's going to give you 4 H pluses. And you're going to times 2 over here, you get 2 H2Os. Right, how do we know that this is reduction? Well, we know it's reduction Because where are the electrons? The electrons are on the reactant side. So it is not being lost, it's not being produced, it's being taken in. So it's gaining electrons. From the videos that I told you to look at, remember Leo, the line says GR, right? GR, right? It's the gaining of electrons is reduction, all right? Gaining of electrons is reduction. I'm going to show you all this in an actual diagram. I just wanted to preface it first with these equations so you kind of know what's going on. So let's start off with that ETC of that third phase of aerobic respiration, and then ATP synthase will be right after this. It'll be very quick though. Okay, so ETC, it's in the, I'll just put this here, it's in the IMM, the inner mitochondrial membrane. Okay, so the inner membrane space is going to be on the inside here. On the bottom, we're going to say it's the mitochondrial matrix. And the other side over here is going to be the IMS, the inner membrane space. So remember that NADH that we talked about earlier? Okay, yeah, maybe I'll put it down here. NADH is over here. It's in the mitochondrial matrix. It's going to enter complex 1, okay? So this here is known as ETC complex 1. This is going to be complex 2, complex 3, complex 4. This is ATP synthase, so that'll be a little bit later on. Let's not get there yet. So this NADH is going to go into complex 1. Why? Because phosphorylation needs to happen, and it happens through oxidation. So this NADH is going to be oxidized. Remember that NADH we just talked about, okay? Remember, it produces how many electrons? It produces those 2 electrons, right? And it also made that NAD plus because obviously it got oxidized, and then we have that H plus floating around as well. So when it goes through this system, it makes sure that the H pluses that are free floating around because of that oxidation gets pushed into the complex and gets released into the other side, right, the IMS. So it drives the H pluses from this matrix to the IMS, and it's creating this proton motor force. Okay, so all the H pluses that are going to be built up here is going to be entered into there. And that high pressure differential is eventually going to go down to ATP synthase, but again, we'll get there shortly. So we're just building up from here and pushing it up to the other side into the IMS. Now there is a Q over here. It's not really in the diagram, but just know that there's a Q here. It's represented as ubiquinone. It undergoes the reduction. Remember, if there's oxidation happening, there is reduction happening. That's what redox meant, right? From that video that you watched. Again, see the video link in the description if you haven't seen those yet. So that Q gets reduced, okay? Oxidation happens, and then that Q gets reduced. That's where it took these electrons. So that taking of electrons from here, from the oxidation, it's going to now turn to QH2. And that QH2, I can't really see it. I'm going to make it green. That QH2 is going to bring those electrons, and now it's in complex 2. Guess what happens here? Remember that FADH2? Let's bring that FADH2 now. That FADH2 goes into complex 2. And very similarly, here, I'll write it here as well. Very similarly, FADH2, like the NADH, breaks up into FAD. And 2 H pluses and 2 electrons. What do you think this is? Obviously, this is oxidation because the electrons are on the right side. It's on the product side, on the right side of that yield symbol, okay? So it's being produced. So this is oxidation. Now, some textbooks don't show it to you this way. I don't like how they do that. This is the correct way. There is oxidation happening, okay? Electrons are on the product side. Some textbooks might just show you just the FADH2 oxidizing to FAD and like hydrogen ions, but that's not correct, okay? There are electrons being produced, given off. So if there's oxidation happening over here, who is going to be receiving it? Obviously, QH2 is going to receive it. So the QH2 is taking and gaining those electrons, and it's going to use that to help keep going to the next complex. Also, it's important to note that complex 2 from that FADH2 that it's interacting with it, complex 2 actually doesn't contribute to the proton gradient, the motor force, pushing the H pluses into there. I know you can see that the H pluses were produced, okay, but complex 2 does not do that. It's only 1, 3, and 4 that does. Okay, so the H pluses go through this one, so the 1. The H pluses will go through the 3 and the 4 as well, but not for the 2. Okay, all right, so technically these two happen simultaneously. So now let's focus on to number 3. Same thing, the H pluses that get built up go through that gradient, and then more H pluses. Okay, so again, H pluses all over the place here. H pluses, H pluses, H pluses get built up, and then they get built up here. They go through 3 and have a lot of H pluses just floating around in here, building up pressure. Okay, lots of positive charges up here now. Finally, the electrons get passed on to cytochrome C. I'll just put that over here, cytochrome C. And again, more motor force to push these H pluses up here. So now there's a big buildup of H pluses all around here, okay, up into the IMS. Where do the electrons go now? Electrons must now bind to oxygen. That's why, like I said in the previous video, that oxygen is the final electron acceptor. So let me minimize this here. So remember this one here, right? The electrons reacts with the final oxygen. And obviously there's a lot of hydrogens around. And due to the electronegativity, they like to bond, and it creates the water. So water is our final product. Okay, so this is why I prefaced it earlier on. The 2 electrons reduce oxygen. Okay, this being reduced. Electrons are on the reactant side. It's interacting with oxygen in order to make that water final product. And obviously water is quite stable, not energetic or calorie dense. So I'll just make a disclaimer here. The 2 electrons now bind to the oxygen with the hydrogen ions to produce water. And I just want you to remember here on just on the side, we do all the electrons in a chain-like fashion, what we just demonstrated in the diagram. So not all at one time. To make it controlled. So in short, it's to transfer energy a bit and a bit and a bit at a time. Otherwise, it's too combustible, too much energy happening all at once, and it can be very damaging to your cells and your body, right? Now, like I said, all the H pluses were up here, right? A ton of H pluses through pressure differential, right? Obviously, you should know that if you have a high concentration on one end, it wants to alleviate that by going to a lower differential area. So then the H pluses now get pushed through, and phosphorylation occurs. Again, we know why it's called oxidative phosphorylation. Oxygen, we just talked about oxygen throughout this whole process. And this phosphorylation, where does it come from? Well, the ADPs that are always floating around then get added with the inorganic phosphates, hence the term phosphorylation, phosphorus, okay, phos. Because right now there is less energy. There's not much energy within an ADP. But with the help of the H pluses, it ensures that it turns this ADP and squeezes it together with a PI into a TP, which obviously has a lot more energy. So I'll put more energy here. How does it do that? Honestly, I don't want to break it down too much, okay? It's not that important for the scope of this course, but just know that this H pluses helps to ensure that the Pi turns into PO43 minus. You should remember from your previous grades, right? PO43 minus, it's a polyatomic ion. It turns that Pi into that PO43 minus, okay? It's a different version, I think it's Y gamma, and it creates a different type of bond where the alpha and beta phosphates are arranged to squish together in this format, okay? A format that is extremely unstable actually, and it has a lot of repulsion. due to the arrangement of those bonds. Very similar to in the glycolysis when we looked at phosphoenolpyruvate. It's in an unstable format. That's what happens with the ATP. So that's why when this P eventually breaks off and makes ADP again, it releases a ton of energy. Okay, all that energy that's built up right now in a potential manner gets released to help you to do work. All right, so what happens at the very, very end? Around 30 to 32, ATP are formed. Much better than what was produced in glycolysis in the Krebs cycle.